Let's now move on. The next speaker is Professor Nagasuma Chandra. We've not been doing long introductions, so I'm just going to skip over the introduction. So I'm sure you'll have a very interesting talk to listen to from her.
Oh, thanks for that.
I think you'll see that she has a very interesting background. She started with B.Pharm, and then she has a PhD from Bristol in 1992 and a postdoc from this very campus.
Okay. Thank you, Karthik, and thanks Chirag and Devnath for this kind invitation. It's also a real pleasure to speak in a session chaired by your former students. Okay. So today I'll be talking to you about multiscale modeling of host responses to infectious diseases. Just to give a brief overview of what we do in the lab. So we do work both breadth-wise and from the systems perspective and depth-wise from a structural perspective. So we do a lot of structural bioinformatics. In today's talk, of course, I'll try to show you how we have combined the two, because we are inspired by this statement by a French philosopher, which, yeah, okay, that's working. Says that the only real voyage of discovery consists not in seeking new landscapes alone, but in having new eyes, new ways to see that. I won't dwell too much about this, but some of the major research areas in the lab is to look at antimicrobial resistance from a systems perspective and structures perspective, and to look at host response to a variety of diseases, infectious diseases on one hand and cancers and other chronic inflammatory diseases on the other. So the global burden of infectious diseases need no introduction at all. We've seen that in the news all the time these days, and we've been pretty worried about that as well. This was just influenza, which I used to show in my slides pre-COVID, because that is very pale as compared to what the numbers that we've been seeing due to COVID, and we don't know how many more pandemics will come or where the thing will take us basically. So it is a major worry, which means that a lot of research has to happen, a lot of attention has to be paid to these things and so on. And especially the viruses as compared to the bacteria, because of the speed with which they infect and then spread across populations. The pathogenic viruses are engaged in an arms race. It's always an arms race, and who wins is decided by multiple things. By the ability of the pathogen to evolve. If the pathogen doesn't do its job of escaping from the immune responses, then it's not a very successful pathogen. So it does that all the time. So it evolves rapidly, and the negative selection, because it's trying to escape some of the immune responses and so on. So for influenza, this is a slide for influenza on the left and COVID on the right, SARS-CoV-2, the causative virus on the right. What you are seeing here are dendrograms, basically phylogenetic trees of some kind. This was based on a serology perspective, how antibodies have evolved. This is based on whole genome sequence comparison, because that became possible and so on. In both cases, we know that there is a continuous antigenic drift from an antibody kind of perspective. There are also variations from a T cell perspective, and we know that the spike protein is important, and both the antibody responses keep changing as well as T cell responses keep changing to these. So what I want to draw your attention to is that all of these are very, very similar to each other. Each strain, each row is a strain here, either here or here. Each row is a strain, and they are placed close together if their whole genome sequences or their serological genotypes are very close to each other. So, and you see them organizing themselves. This is through a standard bioinformatics analysis of comparing sequences and phylogenetic trees and so on. So you see them getting organized into groups, clades, clusters, whatever you call them here, and they're colored based on the time frame and so on. So despite their similarity, there are distinct differences which are lost out with standard analysis of this kind, so you need something more than that. So the host immune recognition itself is a very complex process, multifactorial, number of things are involved really. Just to give you a quick example, for influenza virus it would have to bind to the cells it would, one of the proteins from the viral coat will have to bind to the host cell, and it has to get in, engulfment of the virus, endosome formation, acidification, a number of steps really for the final viral replication and initiating cell death and so on. So one of the key things here, three broad responses that we are all familiar with, and is the innate immune response, and then is the humoral response, or the antibody response, or the cell mediated, or what we call as the T cell response. Okay? So the innate gets kicked off initially, and then it is one of these two, and the time frame is such that this may take a couple of weeks to a month to actually start seeing visible effect of this, whereas cell mediated immunity is a little much faster than that. Okay? And cell mediated immunity is really important in viruses such as influenza and COVID, and also intracellular bacteria such as Mycobacterium tuberculosis. It plays a much higher role than the antibody responses, and there's a huge body of evidence for that. And we actually set out to, COVID being a new beast, we thought we will test it out ourselves, and I think this was during the second lockdown phase, so we were able to get permissions for collecting data and so on by then. So we recruited a cohort of, we collected samples rather from a cohort of COVID patients from asymptomatic, mild, moderate and severe, but not fatal, obviously. None of them died, at least during the time that we were looking at. So they across the disease severity spectrum, and then we did collect their blood transcriptomes, bulk RNA-seq from whole blood. And what you see here is the distinct differences. This is asymptomatic, this is mild, and this is moderate severe. And blue is downregulated, red is upregulated, with respect to healthy controls and so on. There's a lot of change that is happening among COVID patients with different disease severity levels. And I won't dwell on this too much, but we do, this is for a parallel project in the laboratory. So we do model these host responses in a large scale using transcriptomic data and then knowledge-based protein-protein interaction networks to mine out the most important changes that happen in any given condition. And with the same data and the same starting network, this network now is like a road network of a large city like Bangalore. Now you are asking a question, what is different at 8 a.m. versus 12 midnight or something like that, because it's the same network, and over that some dynamic changes are happening, which is what we looked at. And here the time period, of course, is not in hours. The time period, it's not time period really, but it is different categories of people. So in the asymptomatic network for a given T cell response, we see a large amount. Each dot here, each little circle is a gene, and the edges are interactions. The same in mild has reduced, and in moderate severe it's almost this. In fact, it is repressed. We see a lot of downregulation in the HLA antigen presentation and upregulation of the class 2. The T cell arm has two, the T cell response has two arms, the class 1 and the class 2. The class 1 is the cytotoxic T cell response, which is the killer T cell formation and so on. We see a reduction in antigen presentation and the downstream signaling out here, which was interesting. So the open questions now about any of these infectious diseases are that why do some strains cause outbreaks but not all, and why are individuals differently susceptible to the disease? Okay? So we set out with simple objectives to study the effect of host heterogeneity in terms of the cytotoxic T lymphocyte responses on disease susceptibility. Can we map that, can we model that, and then understand who is susceptible and who is not, and by how much, right? And whether we could take this further and to predict infection dynamics in large populations based on this. This is what we looked at. But where do you begin? Whether it's influenza or COVID, and we do a lot of work in tuberculosis as well, you get a large amount of data. Now the data itself can be from the pathogen side, the biology of the pathogen itself. It's also about the host, it's also the clinical data, and very deep molecular data such as protein structures and polymorphisms and a variety of different data, and of course the environmental factors and so on. So certainly, this is not really for this audience, I don't need to convince, but typical biology audience, it would be necessary to show that how biology is changing in the 21st century, why we need to really look at data and then learn from it. So where do you begin really? You know, there's so much data and you have questions which are really broad, but you want to address them. Now, what helps us is the natural hierarchy that is present in biological systems itself. We use both a bottom-up or a top-down approach, or in fact what we would call as a middle-out approach. Start somewhere there and then go both ways, which is what systems modeling is all about. So we did start with the sequence to function models because some of them were well characterized, which I will show you in a minute, and then go all the way up to population level models, which I will talk about. This work is, much of the beginning part of the work is by Sumanta Mukherjee, a former student in the lab who is now heading a group at IBM Research. So he set off to do the following things. First of all, you know, the T cell responses, as I will show you in a minute, involves HLA alleles, HLA or MHC, HLA human leukocyte antigen in humans, or MHC, which stands for major histocompatibility complex in mice. It's the same molecule, and there are thousands of alleles. Okay, and each of us have different alleles, and people actually use it for tracing human migration and large questions like that, because that's very characteristic of an individual. And from that, you know, genome-wide epitope prediction and characterizing host heterogeneity, modeling responses, and simulating infection spread. Roughly this is the flow of what I'm going to show. And now what Sumanta did was to look at the three-dimensional structures of these HLA molecules. And quite a few of them were available in the protein data bank, which is really our main resource for looking at 3D structures. Then we overlaid all of them. This is not new, this was known, but we had to characterize it deeply. And what I'm showing you here is just the binding site, somewhat like a lock and a key, lock being the HLA molecule and key being the little epitope that comes from the virus, a 9 amino acid epitope that comes from the virus. But unlike standard lock and key that you will visualize, this is like a very a lock with a very big keyhole, broad, you know, with really wide keyhole out here, but yet very, very specific. The specificity is so high that if the virus mutates, then it will no longer bind to this one. Just like antigen-antibody interactions, this HLA and epitope which comes from the viral proteins are very specific. What you see here is from the viral protein in stick, and the red thing around it is the keyhole, basically in the HLA molecules, which is surface represented out here. So among these 2000 and odd alleles that we know of, all of them are very similar in their backbone, which means the shape of the lock is the same, but what is different is the shape of the keyhole. A little bit different, not very different, but that's enough to change specificity. And that's what you see here. And this is basically a multiple sequence alignment inferred sequence logo that is shown here, where the height of the letter, this is for the binding site, all these amino acid residues lining the binding site are shown here. We know which ones basically give rise to HLA capability of binding peptides, which ones impart specificity, and so on. A lot of detail is known about these things. Okay. And then again, former students in the lab, Kalidas, Eturu, and Deepesh Nagarajan, who have their own labs at the moment, and of course current students, this is not updated, pardon me, Naren and Santosh Shankar, have developed a suite of algorithms for looking at binding sites. How do we analyze binding sites, not just for HLA, but for all protein molecules together? And using the two things, we asked a question, if we start comparing site to site to site to site in all versus all fashion, we did model the three-dimensional structures of all 2000 HLA alleles and then ask this question. Then we can, just by data, without knowing their function or without looking at literature, we were able to get this kind of a hierarchical classification. They broadly group into 7 subtypes or subgroups, and they further group, and it's a hierarchical classification. Now we wanted to know how correct it was, so we overlay this with the information that is available from a serological perspective, which is how they differentiate between the two, and the colors actually indicate how well they match with each other. More the red, the better the match, and so on. Okay, the more the intensity of the color, red or green or whatever that is, you know, that is, this is programmatically generated. So by and large, the match was excellent, and you do see a very good match with the known things, but of course you get new data. So this gave us an idea. With conventional data, you don't really know why they are similar, but now you have that information. It's no longer a black box. You know why they are similar because you have started with data, 3D data as detailed as it can be, and then you have moved on to it. So these are the keyholes. Now we match the keyholes. Then we ask, if we take the known keys for these keyholes, what do we get? We expect to get something similar, and that's exactly what we see here. This is the same kind of clustering, but now with the keys. The keys are typically 9 amino acid peptides that come from, after a lot of prediction, it comes from viral proteins. Okay, so that is what we see out here. In fact, the clustering of that also different ways. So site-based grouping matches closely with epitope-based grouping, saying that we are okay to proceed further. Now, this is a lot of detail which is necessary if you go into each one, just to give you a flavor. So these are HLA site signatures for different subgroups of HLA alleles, right? And these are epitope signatures for this 9 amino acid, where the second position and 9th position are known to be very important anchor residues, but the positions themselves have different residues. For example, here we have glutamic acid, whereas here we have a leucine. That's because the nature of the keyhole is different in these subtypes. And there is an interactive tool. Unfortunately, website has been hacked recently and we brought it down, but we hope to get it back up again. But there is an interactive browser that you can see. Now, we go back to influenza virus and ditto later for COVID. So we want to look at on this big virus here with so many bases. You know, of course, viruses are not as big as bacteria, but we still want to know which are the regions which can form epitopes. So you take the genome sequence, or RNA or DNA, both are RNA here, predict proteins from that, and then from the proteins we do a bioinformatics prediction of which could be possible epitopes, right? And for example, in this protein out here, we see that this is the epitope, what is in red, this is another epitope, another epitope. When I say epitope, it is for a specific HLA allele, not for any allele, right? It is very specific. And you get a bird's eye view of these so many proteins out there in the virus, which are the epitopes and what is the frequency with which they bind to which allele and so on and so forth. And the epitope prediction itself, there's a lot of work that has gone on. There are very good servers that are available and so on. So they're either based on artificial neural network or combinatorial peptide libraries or energy-based, physics-based methods, simple data-based methods. And usually we end up doing a consensus approach where we probe it with multiple ways and take only the high confidence predictions. So that's what we see out here. Then we asked a question, once we had the predictions, now this is along the sequence position of one protein, PB1 in influenza, and it is colored based on the conservation, extent of conservation among, I think we had 186 different strains for which whole genome sequences were available. So when we compare them, we wanted to ask a question whether the epitope regions were varying much more than other regions in the protein molecule, which means it is under negative selection, right? Epitope regions are varying faster or much more than the other regions in the polypeptide chain. And these are the Shannon entropy plots, which we can see more easily here. See, this is the entire sequence pairwise distance. Each dot here is a comparison of two strains, one protein of two strains, and lots of comparisons are there, which is why you see lots of dots, right? 186 all versus all comparisons are out there. And if there was no difference between the rates of change, you would have seen a diagonal, which you see by and large. But there are interesting ones that you see here where the epitopes have moved on much more than the rest of the protein. In fact, you will see some out here which are really under negative selection. So that was interesting. And again, we need to consolidate. We need to say which allele. These are all allele names, B0702. They're not very friendly, but these are the standard naming scheme that people use. And these are the different proteins that you see out here from the influenza virus. And it says here PB1 has a large number of epitopes for B1503 and so on. And B1503 is the most important allele. If people have that allele, and they are less susceptible to the disease. If they don't have that allele, then they are at a higher risk. Oh, we're out of time already. Okay, let me just race through. Okay, and characterizing heterogeneity in HLA genotypes. Now we have 1884 genes for class, there are 3 loci A, B, C, all put together we get something like 20 trillion genotypes, which is a ridiculous number to compute. But then even if you put all the filters of known genotypes and so on, you get about 111 million genotypes. Now each of us have one genotype because we have 6 alleles expressed in us, which is very unique, right? And we go through all of this data basically, and then make a synthetic population, construct a synthetic population. In the interest of time, I have to run and do an epitope pool in a population, and then we ask a question, can we actually cluster them and then see some interesting things? And indeed that's what happened. When we cluster these genotypes, I automatically grouped into some number of what we call as response types. Now here each response types literally sees the virus in the same way. So all of them basically recognize the epitopes in the same way, and some of them are more susceptible, some of them are less susceptible. So large population, this is about 1 lakh synthetic individuals out here, using real data, we got about few response types, which is the same, and of course we compared it to what was known in literature from seminal work of Doherty et al. And then what we actually see that is these are the pathogenic strains, these are the ethnic groups, and this is the response. That is our predicted response basically. And response, the higher the response, lower the susceptibility, and vice versa. So that's what we are actually looking at here. And some strains actually give rise to low response in all ethnicities, and some strains are good, and some ethnicities themselves are very susceptible to this virus. And we know that H1N1 and things like that have actually emanated from those regions. So it's a very loaded plot. And very quickly, how do we actually look at disease spread? The standard SIR model is what we employ. The S is susceptible, I is infected, and R is recovered. But S now, we wanted to put in host heterogeneity from a genetic information based on the HLA and the model that I just showed you. The S is now divided into S1, S2, S3, and so on. So that's the model. Then we wanted to simulate this, and then let me cut the story short. And then what we get is, of course, the dynamic spread in the population, which actually shows that when it can become an epidemic and when it can't, and so on. Unfortunately, we didn't have this data for COVID until the pandemic was over, but then so we didn't extend it to COVID, but this is a very powerful approach to look at that. Okay. And just to end, I think for COVID, what we actually looked at was, pardon me. Yeah, I'll just stop with this. So we did the same locally. We looked at pressured epitopes, which means which were evolving under negative selection, and asked whether that could explain disease severity that I started with, right? And this was the work of Vishal, who was an undergraduate student and later a master's student in the lab. So just by looking at 6 epitopes, we narrowed down onto 6 epitopes at the end of the day using a variety of filters, and we found that it could separate out asymptomatic, mild, moderate, and severe individuals. Between asymptomatic and severe, there was a very, very significant difference, which showed that just 6 epitopes, 6 regions on this entire SARS-CoV-2 were enough to kind of tell us what would the severity level be, given the HLA genome of these individuals. I think we computed or we inferred the HLA genotypes based on the RNA-seq data of these patients from Bangalore. That's what we looked at. And of course, we did the response typing in the same way, and we see that for different populations, you get, these are the different strains, and these are different populations, example populations, Brazil, Papua New Guinea, and so on and so forth. And we do find even in the same ethnicity, like India, just some data that was available for Andhra Pradesh ethnicity here, how Omicron or Delta versus Kappa actually show differences. We do find a lot of differences out here. With this, let me close the talk. So we can actually model from a large amount of data the disease susceptibilities and understand from more or less a first principles type of approach to predict disease susceptibility. Thank you for your attention.
Thank you, ma'am, for the interesting talk. So I have a question. These epitopes, they are specific to some SARS-CoV-2 region, or they are...
Epitopes come from many proteins, because the spike protein has quite a few, but it's also there from ORF1 and ORF9, 10, which we have not known before. Yeah.
Okay. Thank you.
how poor are we in surveillance, right? I think there was a lot of talk about, Ramesh was telling yesterday about how the UK Biobank had visionary data collection ages ago. And given the kind of work that you had done on H1N1 and so on, I think we were still not in a position to react. I think nobody was in a position to react to COVID, but how else could things have been different? Or is there some retrospective data collection that we can now do to sort of arm ourselves for a future pandemic?
Absolutely, absolutely. It's a very interesting, very important question. So I think we need to have an HLA biobank. It's absolutely important. At least a data bank. We don't need to keep biological specimens, but data is so important, honestly. So I hope the funding agencies will, will hear this. So, and that's so easy to get. All you need is a drop of blood, and there are methods to do it. The sequencing-based methods. There are other methods. We just need HLA data all the time. That's very important. And by surveillance of the virus itself, how it is mutating, I think it has got a lot better after COVID. So we are seeing even from India, from many places, that is being done. But much more requires to be done. It needs consolidation, put into some proper, accessible format without the bureaucracy. I think that's very important. Yeah.
So I have a question on this kind of a universal vaccine that pops up. So in your analysis, does it give you any insights on whether such a vaccine is possible? And one kind of short follow-up is, yesterday there was a poster on multi-epitopes, like concatenating different epitopes together. So your grouping analysis, does that lead to universal vaccine, or should we think about it differently?
Right. I am not that convinced by the universal vaccine thing. First of all, because there's heterogeneity. There's so much heterogeneity. Unless you put many things into that, why would you make it universal? It's only for ease of administration or cost and things like that. But that may be not the most effective. So I think we need to have a hybrid way of thinking, of saying that it needs to be personalized, needs to be customized, but it gets grouped into type A or type B or something of that kind.
So instead of one universal vaccine, you are saying have a short number of vaccines that you then personalize to a group of individuals.
But vaccine itself is such a big field, right? If the vaccine is coming from a host immunity perspective, then what you're saying is right. If it's coming from a pathogen perspective, then a universal vaccine is not unthinkable, because it needs to target those regions that don't change. We know which regions can change and survive, because the virus has to survive too after changing. So we can delineate between the two things.
Ishan.
Really nice talk, ma'am. So maybe I can just summarize what you have. So just to have my understanding correct, so you have, by looking at the HLA locus, you have basically prioritized what epitopes that are changing under selection could be important. Is that what you have done? Is that where the host and pathogen interaction comes into the picture?
Yeah, in this thing, HLA peptide interaction, that's correct.
Okay.
But the HLA, we have like 6 HLA alleles which are codominant in each individual. So we need to consider all of them, was one message that came out. Individual studies, from an immunology perspective, people have looked at one at a time. They've also tried to make associations of HLA with disease in many cases, but that's also one at a time. So what we are trying to say is one at a time is inadequate. We need to look at all 6 at a time and look at the pool of epitopes and not just one epitope, because at the end of the day nobody is prioritizing. There's no process to prioritize when it's actually happening in the cell. All epitopes will be presented and the best of them will win. So it's important to look at the pool of epitopes rather than a
But
single.
given the fact that you know which shapes in the HLA keyhole are also under selection more often, and you know how the epitopes fit.
Keys are under selection, not the keyhole.
The
In
keyhole
our
is not.
lifetime, the
Okay.
molecules won't change.
But do you know, there are these studies at single cell level where they look at T cell receptors and also less like HLA heterogeneity itself.
HLA heterogeneity remains constant in an
Constant, okay.
individual unless there are mutations, whether there are somatic mutations, but that's less of a worry.
Okay.
So what changes is the epitope on the virus, because that's faster, much faster.
So these are two separate things then.
Two separate things.
Okay.
Yeah. T cell receptor is downstream.
Okay. So thanks again to Professor Chandra for a very nice talk. We now have the next speaker. Rukmini Kumar from